Hello, my name is Taeho Jo, Assistant Research Professor at the Indiana University School of Medicine. We have developed a new method for predicting Alzheimer's disease and finding new biomarkers using dual deep learning AI strategy. This presentation will describe the background and result of the method. People are fascinated by the competition between AI and humans. Examples include the Deep Blue chess, AI being in the Jeopardy show, and the victory of AlphaGo. However, the question of who is better, AI or humans, is no longer what we should be asking. AI has advanced rapidly in recent years, and now this question is like humans trying to race against a sports car. Instead of trying to race with cars, it's time to drive it. So the question we should be asking is, what is the best use of AI? There are many answers to this, but the answer we have focused on is the things humans cannot do. This question led us to study the field of neuroimaging and genetics. With neuroimaging that looks similar in our eyes and the genetic information that we can't distinguish, we came to the question, what if AI can predict diseases in advance? That was the beginning of our study. The result was successful. We call this method the dual deep learning AI strategy. And over the past few years, there have been many accomplishments. I will now briefly introduce them. The first was a study of neuroimaging using AI. The background to this study goes like this. Although Alzheimer's disease is not yet fully understood, we know that people with this disease go through brain shrinkage, amyloid plaques, and tau protein tangles. MRIs and PET scans can be used to visualize all three symptoms. With this, we conducted a systematic review of publications that used AI deep learning for neuroimaging data. We reviewed a total of 192 papers from Google Scholar and PubMed and selected 16 studies for further analysis. The yellow line in the figure shows the prediction accuracy. We found PET data to be significantly more accurate than MRI data. At that time, all the PET images were amyloid based. We did not find any studies using tau PET. For that reason, we decided to develop a deep learning based framework for tau PET images. To do this, a total of 300 tau PET scans were collected from Ethne. And I made a 3D convolutional neural network system for this. The framework for applying this method is shown in the figure. CNN, convolutional neural network, is commonly used for 2D images, but I developed and applied 3D CNN to input tau of all brain regions. In addition to the test set, a separate set of independent validations was created and 5-fold cross-validation was used. This table shows the result of the 5-fold cross-validation. We set the training to stop automatically when the accuracy improvement on the test set did not occur up to 10 times. The training was repeatable training to 50 times without overfitting. The average accuracy through these processes was 90.8%. We then applied the explainable AI (XAI) algorithm to identify the most significant parts of the classification. The XAI algorithm we used is the layer-wise relevance propagation. LRP algorithm, which determines how much each pixel contributes to the prediction. LRP algorithm generates relevance heatmaps from the tau PET images in a deep learning-based classification model. This allows us to identify areas of the brain that play an important role in developing Alzheimer's disease. Alzheimer's disease has significantly higher tau deposition in widespread regions, including the bilateral temporal lobes, with global maximum differences in the right and left parahippocampal regions compared to CN. Then, the heat maps of 3D CNN classification were compared to voxel-wise group difference maps. We found that informative regions for AD classification in our result were similar to those found using SPM12. We calculated the AD probability scores of 97 eMCI participants and 71 LMCI participants using the generated model. In this figure, A and B show scatter plots between the Alzheimer's disease probability scores of eMCI and LMCI. The correlation coefficients were 0.43 for eMCI and 0.49 for LMCI. C and D show mean tau accumulation in the medial temporal lobe of eMCI and LMCI. They show that the gap between AD-like and CN-like of LMCI is bigger than the eMCI. The voxel-wise analysis also shows significant group differences. Whole brain voxel-wise analysis in SPM12 was performed, and it shows the differences in tau deposition were more widespread in LMCI compared to eMCI. Next, I'll introduce the second axis of the dual deep learning approach, which is an AI-based genetics project. These two are part of the genetic data from two people. One is a person with Alzheimer's disease, and the other is not. It is nearly impossible to determine which genome is the person with Alzheimer's disease just by looking at this. But we believed that AI could detect Alzheimer's disease, and if these classification predictions performed well, XAI could help determine which areas impact these results most, like tau PET. We can use this as a biomarker of Alzheimer's disease. But when I tried to put this idea into practice, there was a problem. It is known as the curse of dimensionality. This picture shows the problem we had to solve. Although this windmill looks like it has 6 wings, but it's actually 2 windmills built from a different perspective. This means we can misinterpret what's really happening when we ignore dimensions. This picture is from a Nature Method paper. It explains the curse of dimensionality well. In a 2D normal distribution graph of 1,000 points, only 6% are within the standard deviation of x and y equal 1.5. But considering only one dimension, 31% of the points are within the standard deviation of x equal 1.5 and 30% for y equal 1.5. This is what creates the well-known problem in machine learning called the curse of dimensionality. The idea we propose to solve this is shown in this figure. Instead of bringing entire features to deep learning at once, why not input only a portion? To do this, we applied sliding window. It moves over the features. We named this sliding window association test. To determine the optimal algorithms and parameters for the window, several preliminary experiments were done with different algorithms and settings. The final decision was made based on time and accuracy. As a result, the window contains one DCNN with 3 hidden layers between the input and output, and the kernel size was set to 10, input size set to 40. This is how the sliding window method was created, but there are still several problems to solve. To do this, we had to design a 3-step approach. The first problem we had to solve was, even we apply the window approach, the computation time still takes too long. The number of snips after QC was more than 5 million, so moving the sliding window one snip by one would require a total of more than 5 billion times run of deep learning. This led us to start the process of filtering out that were not significantly correlated with a given phenotype. So we made the move of the window by 40 snips on the entire features. By this, all snips were included in the first scan, and through this process, more than 5 million snips were divided into 134,955 fragments. One sliding window takes just 1.15 seconds. So the total time for doing this was just 10 hours. This is one-time process. After running the first step of SWAT on all the features, we found 1,800 fragments with a jet score is higher than the median jet score. This process eventually found that 98.7% of the given total regions were possibly not correlated with the Alzheimer's disease phenotype. So from the next step, it can be focused only on the remaining 1.5%. Now, this figure shows the second step. The task given in the second step was how to select the phenotype associated SNP from the 1,802 fragments. In my previous paper of tau detection, I applied CNN and then applied XAI such as LRP, but at this time I can't use that method because these fragments came from different parts. This means they can't be combined, so I cannot use XAI method. To solve this problem, I started with the idea that the regions before and after the fragments are redundant regions. They are not related to the phenotype. In other words, I set the sliding window's starting point to the number of the fragment size minus 1. By this, each snip of the original fragment will have the same number of deep learning results as the size of fragments. An average of this result of each snip was calculated, and we found that this result correlates with the phenotype. We have decided to call these scores phenotype influence scores. Using the phenotype influence scores, it was possible to draw Manhattan plots and also to identify important SNPs. The third and final step is to create a new machine learning model with a given SNP. Based on the phenotype influence score calculated by second step, the total of 72,800 SNPs in the fragment set were given rankings. We then create a new training model based on the SNPs from the top 100 to the top 10,000. The processing speed of running this with the new data is less than 10 seconds. The accuracy of this experiment is AUC of 0.86. Next, the result. As mentioned earlier, the setting up the sliding window is very important. That is to choose the deep learning algorithms, the parameters, and the window size. The most important factor was, of course, the accuracy, but the running speed was also very important. We experimented with 1D CNN, LSTM, LCM plus CNN, and attention algorithm, which are the most widely used among deep learning algorithms that handle 1D arrays. This figure shows the accuracy of each algorithm. The y-axis shown in blue on the left side is accuracy. The result shows that the accuracies were similar. However, in terms of computation speed, 1D CNN was exceptionally fast. Each fragment took about 0.53 seconds when the 1D CNN was used. The size of the window is also important. Accuracies was similar regardless of the window size, and the speed increased as the window size increased, considering that the in and out time might be affected. We disregarded the windows that were too small and chose 40 as the most appropriate size. This is the result of completing step 2. The PIS scores were all calculated from the SNPs, and using that values, we calculated the Z scores and one-tailed p-values. This figure shows the Manhattan plot where the y-axis is the minus log 10 p-values and x-axis is the SNP position in the genome. The SNP with the smallest p-value was RS42938 in the APOE gene and RS5117 in the APOE C1 gene. The genetic region including APOE, APOC1, TOMM40 gene is known as the strongest genetic risk locus for AD. It is notable that these experiments show different results from the conventional approach using Plink. This is a LocusZoom plot of SNPs within the APOE gene region. The x-axis is the position of the SNP and the y-axis is a minus log 10 of the p-value. Each dot represents the SNPs and the colors represent the squared correlation coefficients with the most important SNPs. This plot shows how our method differs from the conventional methods such as Plink. Plink analyzes only one SNP at a time to calculate the correlation with the phenotype, but our method analyzes nearby SNPs to make a decision. So, where important SNPs are clustered, they are given a higher weight. So when we look at the result of applying our method with LocusZoom, a linear pattern appears. This is very different from the Plink result without the linear pattern. This is the result of the third step. Total SNPs were ranked based on the phenotype influence scores. We made sub-dataset with top SNPs. This figure shows the result 10-fold cross-validation experiment after modeling with 1,000 to 5,000 SNPs. We applied random forest XGBoost together with CNN to compare the classification accuracies, and the highest accuracy was 75.02 from the train set of 4,000 SNPs. The best AUC was 0.8157. So experiments on large-scale whole genome sequencing in ADSP are being prepared. I expect that this experiment will improve our accuracy and allow us to find new genetic variations. This will be an important and necessary step for the treatment of private healthcare. This concludes my introduction to the dual deep learning AI strategy for Alzheimer's disease. During this study, 1 Alzheimer's Association grant, 4 first author papers, and 7 conference abstracts were completed. I would like to thank Dr. Seikin and Dr. No for the overall assistance in making this study possible, as well as co-authors Dr. Researcher, Dr. Bais, Dr. Kim, and all co-authors. Thank you.